This is the start of the skeletal muscle lectures. This will probably be broken down into several parts, so I will title each part. This is skeletal muscle part 1. So we're going to talk about how skeletal muscle works, how it gets its action potentials, and everything else. So let's start with an anatomy review. Review is good for the brain. It makes you remember what you already know. So a skeletal muscle fiber is the fusion of myoblasts. Remember from anatomy, blasts are the cells that divide. Myo means muscle. So during embryonic development, you have this row of myoblasts that the membrane then fuses to make the myofiber. We say myofiber when it's the whole cell. And lots of these cells will do this. Now, these cells will automatically do this if given the chance. I used to grow muscle cell culture, like cell culture, and the cells will automatically fuse and start moving, twitching on the dish if you let them. This gives us the anatomical characteristics of skeletal muscle, which is the cylindrical shaped cell. It is multinucleate because we don't lose all those nuclei for the myoblasts. They just hit the edges. And we see the striations. Now, those striations are the striping. Those are because of sarcomeres, and we're going to look at that structure in a minute. So when we think about skeletal muscle, we have the motor neuron. This is the SE motor neuron that you may remember from anatomy. That motor neuron is from the spinal cord. It goes out to the muscle. And one motor neuron and all of the fibers it controls is the motor unit. So a solid muscle will have thousands of motor units. And one of the things we see is that the finer detail the movement, the smaller the motor unit. So for example, controlling your fingers for writing is a very fine detail motor skill. So your motor units are much smaller, so you can get that fine detail of movement, versus if you think about the muscles that keep your back straight, those have very large motor units because it just has to keep your back straight. It's not doing fine detail work. So one motor neuron and its muscle fibers is called a motor unit. Some reminders about muscles from anatomy again. Muscles always pull, they never push. So muscles move by pulling on the bones. Muscles are paired. We have flexors and extensors, abductors and adductors. The idea is that muscles rarely act alone, meaning that a lot of times a given action is based on more than one muscle, but also muscles, in order to have the reverse direction, have to have an opposite muscle. So two muscles that work together are called synergists. Two muscles that work in the opposite direction are called antagonists. So when I say flexors and extensors, those are what we call antagonistic pairs. Now, the good news is for physiology is that you are not going to be required to name the muscles. We're going to look at how the muscles work, but you don't have to know all the information about the muscles like you did in anatomy. So how do we excite the muscle fiber? So I've drawn this, and this is the cell membrane of the skeletal muscle fiber. And we have this one neuron come in, and we have a point where there's actually an indentation in the cell membrane for what's called the neuro-muscular junction, which sometimes we just abbreviate as NMJ, neuro-muscular junction. And the idea is this is where the axon, the nerve, comes in and connects to the skeletal muscle. And it's usually pretty close to the center of the cell. Here, unlike all those autonomic nervous system where we had G proteins, this is a ligand-gated channel. This time we have acetylcholine, which is what ACH means. And this time we're going to work at a nicotinic channel. This means sodium can come in. Yes, a little potassium can leak out, but for the most part, it's going to be sodium coming into the cell. Well, we know that when sodium comes in, we're going to depolarize this cell. So this is a very fast response because it is a ligand-gated channel. All right, so to continue. When the sodium comes in, we see that it floods this neuromuscular junction, And inside the cell, it's going to diffuse. Well, close by, we have voltage-gated channels. Just like in the axon, the voltage-gated channels will reach threshold and will start an action potential. Now, in a healthy muscle cell, every time we get an action potential, AP in the nerve, we will get an action potential in the muscle cell, and that will lead to a contraction and relaxation. That contraction event is called a twitch. So the idea here is that the nerve, if it is healthy, will always release enough neurotransmitter to cause the cell to depolarize to threshold. Enough sodium will come in to open the voltage-gated channels. And then that action potential, since we're starting in the center, is going to run to each end, of the cell.